Okay, everyone, welcome to final review part 2. Today we're going over chapters 5 through 8. So signaling pathways, microtubules, and cell cycle. Here are the memes for today. Enjoy them. Yes. The slide like that I sent individually.
Yeah, you said like there were videos and
Another battle for another day. I will. Okay, I'll look into it. Of course, there was nothing that could have gone more wrong with the videos and the slides and everything. So obviously technology is not my strong suit, but I'll look into it to see if I can get the links. Okay, so we're starting off with cell signaling and signal transduction. So these are the pathways we're going to be going over. So we have our 4 eukaryotic pathways. The first one was the RTK RAS-MAPK pathway. This is typically signals for cell division. We have the survival pathway, and these are usually cell survival signaling. Then we have JAK-STAT, which they signal for cytokine signaling, cell division, a more variety of things. But in class, we talked about milk protein production. And then we have the GPCR pathways, and these are our most common type of receptor in humans. We have like 800 different versions of these. So we will start with the RTK RAS-MAPK pathway. This is also sometimes called the growth factor signaling pathway. And so just a recap of RTKs. So RTKs are receptor tyrosine kinases. So they're monomers that have an extracellular receptor binding domain, and then they have intracellular tyrosine kinase domains. So in this, we're looking at, in this picture specifically, right up here is our external receptor, and down here is our intracellular tyrosine kinase domain. So the kinase is the enzyme that catalyzes phosphorylation. So I think of it, kinases stick phosphates to things, which we've learned is a good way to activate or inactivate. It's a post-translational modification. And so RTKs are form a dimer. So if you can see here, we have the we have the signal come in. I'm sorry, I'm circling on 2 laptops right now. So we have the signal come in, and the 2 RTK monomers form a dimer. Their tyrosine kinase domains, which are shown in pink here, will interact, and they will simultaneously phosphorylate each other. So we call this autophosphorylation or cross-phosphorylation. And when these are phosphorylated, you can see in this, we have our phosphates in yellow. This creates binding sites for intracellular signaling proteins, so more downstream proteins. So the RTKs are the receptors, and we know that receptors are the beginning of the pathway. So signal binds, they dimerize, our intracellular kinase domains are activated, they cross-phosphorylate the tyrosines on the tyrosine kinase domains, and that is how we begin this pathway. Any questions on the RTKs before we continue on with the pathway? Okay, so then the RTK is going to, well, the next step is RAS. So, or the RAS part of the pathway. So we have this in dark blue here, adapter protein. So the adapter protein is GRB2, that's the GRB2, that binds to the active RTK. And by active, I mean phosphorylated. As you can see, it's binding here at the phosphate. And GRB2 has an SH2 domain. So an SH2 domain is a phosphotyrosine binding domain. So it binds to phosphorylated tyrosines. And we'll see some other SH2 domains in a little bit. So it binds to the phosphorylated tyrosine at the SH2 domain, and it recruits the RAS activating protein, which is in light blue right here. So the RAS activating protein is a RAS-GEF, also known as SOS, son of sevenless. And if you remember, a GEF is a guanine exchange factor, and that causes RAS to change conformations and exchange GDP for GTP. And like I said in the last review session, if you didn't go, GEF does not make GDP a GTP. It causes a conformational shift so that RAS will release GDP and GTP will come in and bind. And this activates RAS. And so once RAS is activated, we continue on to, and RAS is anchored. You can see it shows in this little squiggle because we make lipids look like squiggles. It's anchored to the cell membrane via a lipid. And so we continue on in the pathway, and this initiates a kinase cascade. So this is where we get all of our MAPKs. So RAS activates MAP kinase, which is at the top in green. And so we call that MAPK3. Oh my gosh, thanks for getting this. MAPK3. And so it is important to mention that RAS is not a kinase. RAS does not phosphorylate MAPK3. It activates it in some way that we are not sure about. So RAS somehow activates MAPK3, and a kinase sticks takes the phosphate off ATP and sticks it to MAP kinase, which is MAPK2, which is blue, which then does the same thing to MAP kinase, which is like orange, peach, salmon in this. And so then MAP kinase, well, one, but MAPK will go and phosphorylate downstream proteins that can either change protein activity, or it can phosphorylate transcriptional regulators and change gene expression. So arguably MAP kinase 1, or just MAP kinase, is the most important part of this pathway, in that it just can activate the things that directly affect protein activity and gene expression. Not to say nothing, I don't mean to say most important in that the other things aren't important, but it's just that's kind of the trigger for all the downstream effects. So this can result in cell division. And at the end of the pathway, RAS is deactivated with the help of a GAP. And if you remember, a GAP will come in and help hydrolyze GTP, or help the enzymatic activity of hydrolyzing the GTP to GDP. Any questions on that? Yeah.
I just want to clarify, so at the end of this pathway, it kind of
So so MAP kinase can phosphorylate proteins to either turn proteins on and off, or it can phosphorylate transcription regulators. Well, it kind of phosphorylates all of them. And so transcription regulators could be transcription factors, or like an inhibitor or an activator. In this case, it's a cell division, probably an activator, which then goes and changes gene expression, whether we need to upregulate, downregulate, what we need to do for cell division. So it activates a bunch of stuff.
so let's say it's like
Yeah, that's a great question. So the question was, if GDP can't be switched to GTP, would the whole thing just not work? And if there was no other interference, then yes. There's always the chance that there could be crosstalk, or the cell could find a way to activate the pathway anyway, further downstream. Because if something comes in and activates MAP kinase, it doesn't really matter the upstream signaling, right? But assuming we're looking at the pathway independently, if we never activate RAS with GTP, then we will never have the change in protein activity or change in gene expression. Does that make sense? So it's not a never, because in reality, the cell can compensate sometimes, but in theory, yeah. I don't want to confuse you. Okay, any other questions? Can you see the chat on it?
Yeah, I'll keep going.
Okay, so this is essentially the whole thing together, and you can look at it like a story, and the happy ending is cell growth or cell division. And so you kind of already answered this question of how can I stop the happy ending is, for example, make a RAS that can't bind GTP and stays GDP bound. If we have a RAS that is always inactive, we're never going to activate the rest of the pathway. And essentially, to shut down a pathway, you just have to shut off anything upstream of the result. So if you shut off anything starting at the RTK to MAP kinase 2, if you shut off anything and there's no way to turn the pathway back on further downstream, you're never going to get the results you want. You're not going to have the end result. So essentially, the farther upstream you go, you just stop more things. If we're looking at this, like I said, independently, without any crosstalk, nothing else can affect the pathway. Does that make sense? Good. This is a summary. And I'm not going to read all of it just because this is more for your benefit.
Everything that you said.
So the SH2 domains in this pathway are on the adapter protein, which is, like we said, GRB2. And then there is an SH3 domain on the adapter protein, and we'll talk more about SH3 in a minute, but I would definitely be familiar with what proteins in these pathways have which domains. And so I will be reminding you of the domains throughout this session. And then here's a video where who knows if it'll work when I send it out, but I hope it does.
It should work. I think you have to present it. Like if you put it in presenter mode, I think it works. Otherwise, I don't think it does. If it still doesn't work when you put on presenter mode, let us know. You can like email us. Yeah.
Okay. So the note on this slide is don't ignore anything like the FOXOs and things. There's other classes where you could talk more in depth about that, but just pay attention to what we talked about in class. And this has like BRAF is MAPK3, MEK is MAPK2, and ERK is MAPK. But yeah, this is, if you're a visual learner, this is a great video. And no, it works.
It works.
The MAP kinase.
Okay. Now we'll move on to the survival pathway. So our survival pathway causes cell survival. So the automatic response of a cell is to undergo cell death. So every day, I like to say this, and it genuinely, the cell is fighting every second of every day to stay alive, because it's the default is to just undergo apoptosis. And so the survival pathway inhibits this. And this is really important because this is a pathway that gets messed up a lot in cancer. So if this pathway is overactive and we're not undergoing cell death when we're supposed to, that is a big cause of cancer, is uncontrolled cell growth. And the tricky thing is this shows a really complex pathway kind of situation over here. And you really can turn a lot of things off, and you have to target a lot of things when you do cancer research, which is that's not what this class is about. I'm sorry. I'm taking cancer biology right now. Or cancer genetics. It's fascinating. But yeah, so crosstalk can continue to turn these on even when they're not supposed to be. So survival signaling again starts with RTK. So the survival signal binds to the RTK, and once again, it dimerizes. It does the same thing. And then cross-phosphorylates. And in this case, we have a PI3K, which I'm not going to try to say it, but it's a kinase, come and bind the phosphotyrosine site, which means this also has an SH2 domain, because remember, the SH2 domain is just a phosphotyrosine binding domain. And then PI3 kinase adds a third phosphate because it's a kinase, so it adds phosphates to the inositol phospholipid, which is shown with the squiggly 2 P's here. And that becomes PIP3, which is phosphorylated inositol phospholipid. And that becomes it creates a binding site for downstream proteins. PIP3 is not a protein, it's a phospholipid, so fat. And yeah, it's anchored to the membrane, as you can see by the squiggles, the black squiggles. And so when that becomes activated, becomes a binding site, then protein kinase 1 comes in to bind to PIP3. This has a pH domain, which is the domain that binds the 3 phosphates on PIP3. I think that's unique. I don't think we see any other pH domains. And then kinase 1 also binds a PIP3 with its pH domain. Or no, I said kinase 1, AKT, forgive me. AKT also binds PIP3 with its pH domain. So you can see these two next to each other are both bound to inositol phospholipid. And AKT needs to be phosphorylated by 2 kinases sequentially. So the order does matter. So it first has to be phosphorylated by kinase 1. And kinase 1 will change when it phosphorylates, that changes conformation for AKT, which then reveals the site at which protein kinase 2 can come in and phosphorylate. Once AKT is double phosphorylated, that becomes activated. And once AKT is activated because of the double phosphorylation, it dissociates from PIP3 and it enters the cytosol. So it's all bound, and then once it's active, it's released and it continues on its way. And then P10 just is a phosphatase that takes the phosphate off of PIP3, so it doesn't stay active forever. And AKT goes into the cytosol and binds to BAD. So BAD is normally active and inhibiting BCL2. BCL2 is the apoptosis inhibitory protein, which means it inhibits apoptosis. So BCL2 inhibits apoptosis. So obviously, if we want our cell to survive, we want BCL2 to be activated. So BAD, when it's not phosphorylated, is inhibiting BCL2, which is inhibiting the inhibition of apoptosis. So AKT essentially comes in and phosphorylates BAD. This inactivates BAD, and BAD, which is in blue here, when it's phosphorylated and inactive, it releases BCL2, our active apoptosis or apoptosis inhibitory protein. And BCL2 can now relay the signal to stop apoptosis. So yeah, it does that by binding to the mitochondria. You don't need to know more than that. I know that's really confusing because it's a little different than what we've done in the past where everything just turns things on. So any questions on BAD and BCL2? Okay. So this is a little chart or little just simplified version of what I was saying. That might help simplify that. If we have BAD and it's active, it's bound to BCL2, which means BCL2 is inactive, which means there's holes in the mitochondria and we die. The cell, not us. But then we phosphorylate and inactivate BAD. It releases BCL2. Active BCL2 can inhibit apoptosis and the cell lives. So this is just another picture of the whole thing, starting with the phosphorylation of AKT. Yeah. So this is the summary. Again, if you want to go back, it's everything I just said. And then here's a big picture of all of it. It's not complicated at all. So any questions? And mTOR in this kinase 2 is called mTOR. That's another name for it. So are there any questions on the survival pathway before we go to the JAK-STAT pathway? Okay, so we'll do JAK-STAT. So we will discuss if JAK-STAT is considered to be an RTK and how the pathway works. So JAK-STAT is involved in many processes, like we said. So there's cytokines, so the immune system, there's cell division, there's milk production or milk protein production. So the JAK receptor is not an RTK. That's really important. So it's a receptor, but it does not function as a kinase. It doesn't have a kinase domain. So it has kinase subunits that associate with it. So you can see in this little diagram, our JAKs are I'll get there. I'm going to keep talking and then I'll show the whole thing. So yeah, it has kinase subunits that associate with it. And then JAK itself is also not an RTK. So the receptor is not an RTK and the protein itself is not an RTK. There's no autophosphorylation. And JAK stands for just another kinase. It's a non-receptor tyrosine kinase. So this is in the case of making milk proteins. So prolactin is the receptor that comes in. It shows this in red. And then we have the JAK subunits phosphorylating each other, which then phosphorylate. Let me do it in words and that may help. So JAK 1 and 2 are bound to the receptor. So those are the blue parts. So the prolactin signal comes in and the receptor dimerizes. So similar to RTK in the dimerization. This activates JAKs cross-phosphorylating each other. So the JAKs phosphorylate each other and then they're activated. That allows them to then phosphorylate the receptor. So the receptor itself is not phosphorylating the other receptor. It's these JAK subunits that are phosphorylating once they're active. And then once those are active, they have it creates an SH2 binding area because it's a phosphotyrosine. And then STAT comes in this case, they're pink. So we have STAT, and in this example, we have STAT5. So 2 STAT monomers come in and bind via their SH2 domains. So this is another example of a protein with an SH2 domain. And STATs get phosphorylated by the JAKs too. So the JAKs are kind of phosphorylating everything. This causes a conformational change so that the STATs dissociate from the receptor and then they form a dimer using their SH2 domains. And so then it's this little 2-STAT protein here. And this is allowed to then enter the nucleus and it acts as a transcription factor. And as we know, for nuclear import, it uses important. So the important thing here is that to realize that the SH2 domain allows STATs to bind the phosphotyrosine on the receptor, but then they also have phosphotyrosines on themselves so that they can bind each other using the SH2 domains. So that's a little confusing because JAKs are kind of phosphorylating everything, but JAKs are technically not the receptor. They're a kinase subunit. Does that make sense? Do we have any questions? Okay. So this is a summary. So this one's a little shorter. We don't go into as much. But yeah, the most important thing to know is that JAK is not a tyrosine kinase. It is not an RTK. The subunits phosphorylate each other and those phosphorylate the receptors. And then the SH2 domain on the STATs. Any questions on this overall before I move on? Okay. This is just another picture, close up. And the last pathway is the GPCR pathway. This stands for G protein coupled receptor. G protein coupled receptors are the most common receptor in our bodies, like I said. And we're just going to talk about all these things. So here's a picture that we've looked at in class, and it's kind of blurry and not great quality, but we'll blow it up. So GPCRs are a 7 transmembrane domain structure receptor that is, and they're alpha helices, which you can't see great in this, but yeah, they're alpha helices. And their ends are put in the hydrophilic membrane, and the middle of their domains are hydrophobic, which makes sense. And this chemically makes them stay put in the membrane due to the hydrophobicity, which I don't even know if that's a word, but I wrote it down. Yeah, so that's the structure. And it is bound to a G protein made up of 3 subunits. We have the alpha, the beta, and the gamma. In this case, our alpha is blue, our beta is an orangey red, and gamma is green. And the alpha subunit is a GTPase. So remember, a GTPase is going to hydrolyze GTP to GDP. So in the inactive state, G alpha, which is the GTPase, is bound to GDP. A ligand will bind the receptor, the GPCR, which I'm probably just going to call the receptor from now on because it's a mouthful. And this causes G alpha a conformational shift. And it causes G alpha to let go of GDP, which we've seen multiple times, right? Is something letting go of GDP. And then GTP can diffuse in because, like we've said, GTP is in much, much higher concentrations in the cell. So it diffuses into the G alpha, and that activates G alpha. This results in G alpha dissociating from the G beta and G gamma complex. So G beta and G gamma are always in complex together. They do not become separated. And so that complex can then go bind an effector. Both complexes can go bind effectors and start a cascade. So G alpha is now active. It can bind its own effector. Oh, well, yeah. So they both bind effectors and start cascades. I put that in 2. That's weird. The 2 bullets. And then they're doing that, but obviously we don't want this to stay on forever. So the deactivation of G alpha occurs when RGS binds to it. And RGS is similar to a gap. Remember, a gap comes in and helps, oh gosh, what's the protein? Why am I struggling?
RGS helps the gene.
No, I'm saying gap, import and export. Why am I blanking? What does gap help with? What's the protein involved in import and export, the enzyme? Is it a RAS?
An import.
Huh? RAN, not RAS. Okay. Sorry, I totally just had a brain fart. Yeah, so like GAP comes in and helps RAN be a better enzyme. RGS binds and helps G alpha become a better GTPase. Yeah, sorry, wasn't clear about that. So this accelerates G alpha's GTPase activity and causes it to hydrolyze GTP to GDP faster. Then G alpha, G beta, and G gamma all come back together, forming the GPCR complex that we started with inactive. So the GPCR is like GEF, which GEF is the guanine exchange factor that causes a conformational shift. So you release GDP. And then RGS is like GAP, where it helps your GTPase become a better enzyme. Does that make sense? Even though I had that pause there. I forgot what RAN was for a second. Okay. So this is just a different picture and kind of shows the cycle. I'm not the hugest fan of this picture, so we'll go on to the next one. This picture is the one that you would see if there's a question in which Laura decides to give this to you, but this is probably my favorite. And then common misconceptions are the gaps and the gaffs. is that they're not enzymes. And I said this last time and I'll say it again. They are not enzymes. They just, GEF changes conformation and promotes GDP to dissociate, and GAP binds the GTPase to accelerate its intrinsic enzymatic activity. GAP does not hydrolyze the GTP. Look, RAN, nuclear import export. Yeah, so these remember that regulation signals and pathways are highly regulated. So there's activation and deactivation. So you don't have too much signal or too little signal, because like we talked about, that causes problems such as cancer or a plethora of problems. And just mechanistically, just make sure that you know all of the proteins and their functions and kind of what they do. And obviously there's some excess proteins like FOXO before. If you watch a video and it includes that, ignore it. If we didn't talk about it, you don't need to know it. Oh, any questions on any of the pathways before we move on? I know that this is a dense section. My piece of advice would be to draw them out. I have my journal in front of me and I drew these out
Yeah, I was
like
gonna say the
17 times.
Same thing. If you haven't done so already from like the previous exams, like you can just bring it again. I would suggest redrawing it just so like you have it like fresh in your memory again. But just even like having the drawings with you, because you are allowed to have the cheat sheets.
Can't relate.
Yeah, so that you are allowed to have the cheat sheets, it's just like a little easier on you. Like if she asks, oh, if I don't know, if like the GPCR receptor is non-functional, then what happens? And then you can just kind of like follow your pathway and see like what turns on, what wouldn't turn on, things like that.
I'd also put in your domains and what has what domain. Know what has an SH2 domain. I don't know, in my gut, I just feel like you need to know that because I wrote that down a lot of times. Yeah. Yeah, there was SH3 somewhere and I don't know where it went.
It's not as common.
SH3 is not as common. So an SH3 domain. Oh, so the G, I don't think that you did. There's a part of the G coupled protein receptor that we went through our year that has the SH3. So an SH2 domain, all an SH2 domain means is there's a portion of the protein that has a specific structure that can bind to phosphorylated tyrosines. And that's called a phosphotyrosine domain. So that's an SH2 domain. And so anything that has that domain can bind to a phosphorylated tyrosine. And so that's used a lot in these because, as you can see, these receptors have phosphorylation at their tyrosines. That's involved in the first three pathways. So RTK and also JAK-STAT receptors. So that's an SH2 domain. And so in the case of STAT, which is here, sorry, I'm not back to the right slide on this computer. So in the case of STAT, we recognize the tyrosine that's phosphorylated on the receptor, but they also, when they're phosphorylated at a tyrosine when we activate the STAT and phosphorylate it, which causes them to bind at their own SH2 domains to each other. So it's a little convoluted. So that's all that is. And then an SH3 domain.
So this would be like an SH3 domain right here.
Yeah, so there's an SH3 domain on, where am I?
Slide 10.
I don't have numbers.
Oh, sorry.
Okay, so there's an SH3 domain on the adapter protein. So an SH3 domain, I don't believe you went too far into it, but an SH3 domain has an internal PXXP motif sequence. Did you talk about SH3 at all this semester?
Yeah, that sounds familiar.
So, that's not the domain. The domain binds at a PXXP domain, and XX means that it can be anything. It just has to be the same too. And so in the case of, I'm trying to think what see, we did chronic, you didn't do chronic leukemia, right? That's where we mostly did SH3, but yeah, so it just, SH3 is less common. And in this case, I think we only see it in the adapter and the RAS activating protein between each other is just that PXXP motif, which is just saying that sequence, the SH3 domain binds there, and that's where the adapter protein binds.
So I just wanna clarify, so the adapter protein
Yes.
That has
So
SH2.
in what we've gone through, the things that have the SH2 domain are the adapter protein, PIK3, STAT. I think that's it. I have my list that I lost somewhere. Yeah, so PI3 kinase has SH2, GRAB2, which is the adapter protein or activator protein, or it has many names. And then the STAT are the ones that have SH2. And then, so the adapter protein, when I say that it has an SH2 and an SH3, the SH2 is binding the receptor, the phosphorylated receptor, and the SH3 is connecting with the RAS activating protein. So different spots. Okay. Any other questions? That's a great question. I'm gonna be annoying and not. Okay, so we'll move on to my favorite, which is microtubules.
Oh, that's not supposed to say Syed. Oops.
Oh, cool.
He's not here.
Syed's not here. He
He
graduated.
graduated.
So microtubules are, we're gonna talk about the spindle and spindle assembly and microtubules. So we have 3 types of microtubules. And the spindle is formed out of microtubules. So we have asters, which in this case are pink. Actually, the asters are green. I don't know why it's color coded pink, but the asters are green and they grow from the centrosome and attach to the cell cortex. So those are the ones that are going outwards towards the cell cortex because obviously our chromosomes are lined up in the middle. So this is the outside. Then we have our kinetochores, which grow from the centrosome and attach to the kinetochore. So those are blue. And so you can see they're growing from the centrosome still and coming to connect at the kinetochore, which is just a part of the chromosome where those bind. And then we have interpolar microtubules, which grow from the centrosome, shocker, and overlap at the spindle. And that's these pink ones with the little bead looking things, which are kinesins, dyneins. What are they called? Kinesins, dyneins, motor proteins. I've gone through this too many times. And they can grow and shrink. And so our microtubules are made up of tubulin. And tubulin is a dimer with 2 subunits. So it has its alpha subunit and its beta subunit. And remember, a subunit is just an individually folded polypeptide. So the alpha subunit has the GTP binding pocket, but the GTP is buried. So the beta subunit has the GTP binding pocket that's exposed. And so it can hydrolyze the GTP because it has more GTP regulation. So our beta subunit is the one that hydrolyzes our GTP and has the enzymatic function. So the minus end is the exposed end of the alpha subunit. So it's labeled here, alpha is blue, and there's less growth on that end versus the plus end, which is the beta exposed subunit. And remember that the minus and plus end do not have anything to do with charge. It is only relating to the fact the plus end grows and shrinks more or quicker and is more dynamic than the minus end, which is slower. So there's no charge involved. It's just talking about they're not equal. They're not symmetrical. So the structure of the microtubule is made of these tubulin heterodimers, and they're polar. Like I said, they're not equal. And so for the most part, microtubules are hollow tubes with 13 protofilaments, which are just lines of tubulin heterodimers. And the 13 make kind of a hollow tube. And microtubules originate in the centrosome. The minus end is often embedded in the microtubule organizing center, which we'll talk a little more about. And the plus end extends outwards. So while the minus end shrinks faster, usually in vivo, which means in a living cell, it is attached to a microtubule organizing center. So it's not actually having any dynamic function. So this is now in vitro, which means in the lab. Well, in vivo in the cell, no shrinkage at the minus end or growth or shrinkage, like I said. So we have, in the case of humans, gamma tubulin ring complexes in the centrosome. And you can see that in this picture. But in vitro, we can throw pure tubulin with GTP in a test tube. And the pro is that this is a controlled environment where we can kind of look at microtubules and their what they're doing, and we can throw whatever we want in there to really control our different parts of the experiment. And that, but our cons are obviously, if we do it in a petri dish, we don't know if it's actually going to work in a living cell. So we're going to go through some of the experiments. So microtubule nucleation in vivo, again, in vivo in life. So these are in living cells or in like natural cells, not in the lab. So the main idea is these gamma tubulin rings are formed and act as the base to start microtubule growth. And this occurs at the centrosome. And we remember talking about the fact that we need these nucleating sites because it just takes so much tubulin and time to create a microtubule from scratch that is just not efficient. And also, like, these are involved in spindle formation, and the spindle doesn't form out of nothing. I mean, it kind of, well, yeah, so it forms at the centrosome to make the spindle. And so here's a pretty picture of our gamma tubulin. And then looking at dynamics in vitro, I know that you had to draw some pictures on an exam, and I know you had to draw some pictures for problem sets. So these are the pictures, and this is a time course experiment using I'm not even going to try. I'm not great with words and like pronouncing things. But this is a graph of absorbance versus time. So the more microtubules present, the higher the absorption. That's essentially what you need to know for this. And so as our curve increases, this just means we have more microtubule. So we have three phases that we typically see. We have the lag phase, which is the beginning where the tubulin dimers become oligomers, which become protofilaments, which then become sheets of protofilaments. And then you have to close the tubulin, and then you can start elongating. So that's what's happening during the lag phase. And that's why it takes some time to form the beginning of the microtubules. Because in this case, we didn't throw in anything to grow off of. Then two, we have the elongation phase. So at this point, it's easier for microtubules to add tubulin because we already have our base. And this is where our rate of addition is the fastest. So this is where our slope is the steepest. And then we have our plateau phase, which is where we reach equilibrium of microtubule addition and microtubule depolymerization. So the tubulin concentrations drop, and the tubulin is hydrolyzing GTP to GDP at this point. So we'll start with the lag phase. Like we said, the formation of seeds, we call them from scratch, is called de novo, which means from nothing. And wow, I just went through all of this. I forgot I had a slide on it. So the higher the concentration of free tubulin, the shorter the lag phase, which makes sense because if you have a higher concentration, it's going to be easier for things to bump into each other and build this seed, versus if things take longer to find each other. And then so what if stable aggregates or seeds, a question?
Yeah, it's coming.
Oh, what if stable aggregates or seeds were provided or already existed? Like if we threw in gamma tubulin rings in the cells, what do you think would happen? Or something like the gamma tubulin rings in the cells? Yeah.
Very short lag phase.
Yeah. So the answer was a very short lag phase, if a lag phase at all, right? Like, I mean, there probably would be a bit of a lag phase, but yeah, the lag phase would definitely shorten because we don't have to build from nothing because we have something. So we can just start elongating. Awesome. Then we have our elongation phase. So we start with the small microtubule seeds at the bottom, and we start rapidly adding the tubulin, primarily to the plus end, like we said, because that's the more dynamic side, and that's where the beta tubulin is exposed. And so GTP bound tubulin has a greater affinity for, like, the tubulins have greater affinity when they're GTP bound, which is why it adds faster to the plus end, because remember, the plus end is where the GTP is exposed. And then as microtubules elongate, your concentration, that's what those little brackets mean, of free tubulin will decrease, obviously, because unless we're continuously adding the tubulin, we run out of free tubulin because we're adding it to the microtubule. So your rate of assembly is going to have to start decreasing because you have less and less tubulin to add on, which takes us to the plateau phase, which is where our assembly equals the amount of deassembly, or the rate of deassembly, because GTP hydrolysis is always happening. And so eventually the GTP hydrolysis is going to catch up. So there's no affinity between GDP bound tubulins, and this is what's causing the depolymerization or disassembly, whatever you prefer to use. And hydrolyzed tubulin can't bind again because in our test tube, remember, we can't just suddenly make GTP. We need a way to exchange the GDP for GTP, and we don't have that. So we can't rebind, and eventually all the tubulin will be GDP bound. And so this is not static, just that the polymerization rate is equal to depolymerization rate now. So like, it's not just like the microtubules don't just stop. Like the polymerization and depolymerization are still happening. They're just equal to each other, which is why the levels of microtubules are not changing. Until hydrolysis is going to catch up in the closed system, and all the tubulin is going to be GDP bound, and catastrophe is going to happen. So our microtubules are going to fall apart because we don't have the GTP cap anymore. And so I know there was a question on exam 2 that was at, like, after a short period of time, you're going to see the plateau phase, right? But after a longer period of time, our graph is suddenly going to drop, right? Does that make sense? So our graph, do you want to draw this? Our graph is going to go from plateau to sharply decreasing because we catastrophe. Because we can't polymerize, we're just going to continue to depolymerize because we ran out of GTP tubulin. So, oh, wow. So what happens after hydrolysis completely catches up? It catastrophes. What's the way that we could cause the microtubules to polymerize? What could we add? Maybe. Any ideas? Yeah, so if we add more GTP bound tubulin, we can continue growing, right? Because we couldn't exchange the GDP, but we can add more GTP tubulins. What's a way that we can cause shrinking? There's several answers, so whatever.
Oh, Taxol?
Yeah, we'll get to the drug. So Taxol, we could also like cut the microtubules in half, right? Because we expose the GDP end. Remove the GTP caps. Anytime there's GDP bound tubulin exposed, we're gonna start depolymerizing, right? Because we don't bind the GDP end because the affinity is for the GTP end. Any questions on those? Okay.
Removing GCAP, cutting in half, and then like, what was that for, sorry?
For causing depolymerization. Okay. So the dynamic instability I've been talking about, and this is whether the microtubules are growing or shrinking at any given time, and that's dependent on the rate of hydrolysis versus the rate of addition. So if hydrolysis is happening more quickly than addition, we're shrinking or depolymerizing, and this is unstable. But if the rate of addition is greater than the rate of hydrolysis, we are growing, and this is more stable because we have our GTP cap. So this is a great picture showing that. And We can save a shrinking microtubule, like we said, by adding more GTP bound tubulin, but catastrophe happens. I'll get to it. Actually, it's on a slide. So this is a good video. Yeah, this is a good video if you want to watch it.
Welcome to
No,
Please.
that one works.
Welcome to Cell Clips. In this video,
Okay, and then here are the drugs, like you said, Taxol. So we have 3 drugs that we focused on. So Taxol binds and stabilizes microtubules, which means there's no depolymerization. And if our microtubules get stuck and can't depolymerize and get stuck forever, it's used as a chemotherapy because obviously, like if you keep your spindle forever and we're not getting rid of it like we're supposed to be, they're not shrinking to go into anaphase, our cells gonna freak out and it's eventually gonna undergo apoptosis. So that's why it's used as a chemotherapy, a very common one. Then we have colchicine, which does the opposite, is that it's a microtubule destabilizer. So it binds the subunits and prevents polymerization. And this one is used to control inflammatory responses. And then vinblastine is similar to colchicine, which it prevents polymerization by binding the subunits. It destabilizes the microtubule, and it is also used as a chemotherapy because if you have too many microtubules, we don't like that, but also we do need to form them. And if we don't form them, we're also going to undergo cell death. Any questions on those? So Taxol stabilizes, colchicine and vinblastine destabilize. They're not better or worse because in both cases it's extreme and we use it to kill cells. Okay. So if you add colchicine to your in vitro assay at time equals 0, what results are you expecting to see? Any ideas? No microtubules, correct. If you add colchicine, it prevents binding of subunits. So if you add it at the beginning, before you've made anything, you're not gonna make anything, right? Because you can't bind your subunit. So you're not gonna be making anything. Okay. Oh. Yeah, so there's your little graph drawn. And then the last part of this is microtubule associated I don't know why I just had like a breakdown. Microtubule associated proteins, also known as MAPs, and motors. So our microtubule associated proteins, the example that we talked about is tau. So a tau is a protein that is highly expressed in the central nervous system, in our neurons. So this is brain stuff. And so tau binds to microtubules and stabilizes them. So this tau is pretty much a natural Taxol. I mean, they're not the same, but similar to how Taxol stabilizes, tau is also a stabilizer. So when we have dysfunctional tau, this causes neural detriments. And so examples that we know are dementia and Alzheimer's, because hyperphosphorylated version of tau can no longer bind microtubules. So if tau is forever phosphorylated, it can't bind the microtubules like it normally did. And then it freaks out and aggregates with itself, kind of like how hydrophobic R groups will aggregate when they're exposed. Connections. But so tau is going to aggregate and it makes these big clumps and large tangles, and that's why you see the kind of degeneration, and you'll see like in a brain, an Alzheimer's or dementia brain, you can see the effects of tau. I don't know if we have a picture, but so yeah, tau is a microtubule associated protein because we need it, and it's associated with microtubules and stabilizing them, but obviously it itself is not a microtubule. So yeah, that's it on MAPs that we'll do. And then, okay. And then motors, the last part of microtubules. So motor proteins hydrolyze ATP to move along the microtubule. So these are our little feet that walk. So motors can do a bunch of fun stuff. So they can transport cargo around the cell using the microtubules. So they walk on the microtubules, like the microtubules are the highway or the road, and the motors are the car. And the cars can carry you. So say you're the cargo, or it can carry other stuff, you know, when you put a bunch of stuff in your car. Proteins have, these motor proteins have directionality. So we have kinesins. Kinesins walk towards the plus end of the microtubules, except for kinesin 14, which walk towards the minus end. But for the most part, kinesins walk to the plus end. And kinesins are very diverse, and there's a lot of different families of kinesins that can do different things. And we'll talk a little bit about some of them. And then we have dyneins that walk towards the minus end of the microtubule. And these are common in what you'll see in the flagella and cilia, which are your little hairs or your little tails that help different things move. So opposing forces from the motor proteins, our main idea here is the spindle formation requires different opposing forces from kinesins and dyneins, or that are a result of kinesins and dyneins. So they walk towards opposite ends of the microtubule to create opposing pulling forces. So EG5 is an example of a motor protein that binds interpolar microtubules that overlap. So remember, we looked at the pink ones you can see in the top right here. Why is it doing this? These little balls that are connecting those interpolars, those are EG5s. So they overlap, or they bind to those, and it's a special kinesin. It is a plus end directed motor, and it attaches to these microtubules to push the poles apart. So EG5 is what kind of kinesin if it has two subunits on both sides? Do we all agree on that? It's a kinesin 5. Everyone looks like they're doubting themselves. Yeah, it's a kinesin 5. Confidence, you got it. So kinesin 5 have two motor domains on either side. And so I'm jumping the gun a little bit, but you can see this picture down here. So it attaches to two microtubules, and they're both walking towards the plus end, which is resulting in the microtubules pushing the spindle poles apart. And this is what ends up leading the cell into anaphase, right? Because we need to pull our chromosomes apart. And so monostrol is a drug that it inhibits this, and the spindle collapses. So that's bad. Yeah, so the basic role of the kinesins and dyneins in the case of the spindle are to push the poles apart and separate the chromosomes. But obviously, like we said, they do all kinds of stuff like transporting stuff around the cell, which you can see in this kinesin moving this vesicle. Let's pretend the vesicle has something important in it. Okay, so I just answered this. I keep doing this and I'm really sorry. I read the questions beforehand and I keep answering them. How does kinesin 5, EG5, push spindle poles apart? I liked that. I'm sorry, you can't see my picture. Can I turn on the video on this for just 1 second so I can do that?
Yes, of course.
The answer to that question was, okay, I'm done. So yeah, so they bind the microtubules. No, that was great. Don't be embarrassed. That was amazing. I needed that. So it binds two separate microtubules, walking the plus end for both, which is opposing directions. Yeah, and it pushes the spindle poles apart, and that results in pushing the spindle poles apart. So, and then just the kinesin families that we talked about, the important ones. So we have kinesin 1. Sorry, the Zoom is being a little slow. Tina's laptop having a breakdown.
Yeah.
Okay, so kinesin 1 are dimers that walk along the microtubules, and they're mostly just transporting cargo around the cell. And then we have kinesin 3, which is movement of synaptic vesicles, which is transporting cargo around neurons. Because synapses happen at neurons, but that's not super important. And then your kinesin 5 are your tetramers that have the two motor domains on the head and the tail, which cross-link the microtubules and add the stability. And EG5 is an example of this. And the kinesin 5s are required for proper spindle assembly. And then you have kinesin 13. So these do not walk on the microtubules like the other ones. An example of this was MCAC, which is all caps, MCAC and cell division. So they can walk in either direction and they destabilize the plus ends of microtubules. It uses kinesin 13s use ATP to pluck off tubulin monomers. And I love the word pluck. I don't think I put it on here though. So yeah, during anaphase A, they shorten kinetochore microtubules. So yeah, they just destabilize the plus end. So they're a fun one. Any questions on our special families? And then we got dyneins, which are just multifunctional minus end directed motors, which goes towards the centrosomes, right? Because that's where our minus end is. And this is kind of a confusing picture about kinesins and dyneins and arrows pointing to their direction they're going. And they're holding fun things.
We forgot to put the slides on, or I forgot like the picture, but there's like this one picture for opposing forces that I think is a good one. It's like a white, like old looking printed picture, and it has like, they look like bunnies that are like walking, if you guys know what I'm talking about. I can like add in the slides too when I send them out to everyone later, but it's a really good picture that shows like opposing forces.
I don't know what she's talking about, and
I saw
I'm
someone
excited to see it. I'm excited to see it later.
It's like, I don't know, they look like bunnies.
Okay.
It's just,
Well,
yeah, I feel, do you think it's helpful? Okay.
Okay.
Cool.
Well,
I'll
I'm
add it in.
happy to see it. And then here's a fun video. I love watching. I don't know if anyone's seen this. I don't know if I'm just on a very specific side of TikTok, but there's the sound that's like, let me see you walk, 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 walk. And it's with kinesins walking along microtubules. It's a good time. This video is not that exciting, but this is a good video that shows the motors walking.
We replace this one with the TikTok ones.
I tried to find it. I spent like 10 minutes in class one day trying to find this video that I saw on TikTok. I couldn't find it and I'm so upset.
Yes, I TikTok.
I sent it to the group chat when I found it. I didn't find the right one, but I found one. Maybe I'll send it out later to the whole class. That's abuse of my power of the email. Okay, but this is a less fun version, but a very helpful video of kinesins walking. And on that note, we're going to move on to the cell cycle. And you get to listen to Tina talk now. Any questions on microtubules before we move on? No. Oh, chat.
I want to be on that side of TikTok. Yeah.
Yeah.
Me too.
Someone said in the chat that they want to be on that side of TikTok, and like I think we should all be on that side of
Get me
TikTok.
outside. Do you want to switch things?
Yeah, can we switch? Can you do both for a second? I
Yes,
have to go
got
back.
you. You're good. I got it.
Where's the closest pad? I'll find it.
Okay, so we're gonna go into cell cycle now. So starting off with just like a little basic overview, you have G1, G0, or G0, S phase, G2 phase, and M phase. So we'll obviously go into each one of them a little bit more specifically, but you start off with G1. That's obvious. That's like the start of the cell cycle, your first gap. So G for gap or growth, however you want to think about it. That's when, you know, the cell's preparing its DNA, it's growing, it's making sure that it has enough nutrients, it's got all the organelles it needs, all that jazz. This is also where it has the restriction point. So that that checkpoint, the restriction point, like the point of no return, that's where it commits to the cell cycle. You could also have the G0 phase where it's non-dividing. So it doesn't necessarily mean it's doing nothing, but it just means that it's not necessarily in the actual process of the cell cycle. And then you have S phase, S for synthesis, as I like to think of it. I don't know if like these are actually what the letters stand for, but these have always helped me. So synthesis, you're synthesizing DNA replication, end of the phase checkpoint, your G2 phase or your second growth phase, that's when it's also growing again. That's the whole like We1 scenario where it prevents the cell from going on so that it can grow a little bit more and just like make sure that it's ready for segregation. And then your lastly M phase, which has its own different phases within M phase, your P phase, M phase, A and T. And then it also has the metaphase checkpoint. So that's where like a lot of the action's actually happening. Thanks for coming.
Okay.
So your CDKs are the master cell cycle regulators. They are present and very present throughout the entire cell cycle. They are the master cell regulators because of how present they are. So if you restrict any of the CDKs from happening at any point within the cell cycle, that can prevent CDK, or that can prevent cell cycle from continuing, and it'll stop the cell cycle if needed. They're regulated by cyclins. So depending on what cyclin you have, that's going to determine whether or not your CDK is going to be active. So that's why it's cyclin dependent. You can't have an active CDK if you don't have cyclin along with it. It's another way to regulate CDK. So there's a bunch of different ways to regulate CDK is by phosphorylation or dephosphorylation. So again, like a lot of times, I think a lot of students have the misinterpretation that phosphorylation means good and then dephosphorylation means bad. That doesn't necessarily mean the same thing. So you can phosphorylate or dephosphorylate. It's not good or bad. It's just all dependent on like what the cell needs. So for example, We1 phosphorylates, but it's like an inhibitory one. And then lastly, it can also be regulated by CDKI, so CDK inhibitors. And it does exactly what it says. It stops the CDKs from functioning, and thus it stops like the cell cycle from functioning if needed. Yeah, so point blank, it's the master cell regulator. Throughout the entire cell cycle, you can target these CDKs and then target specific CDKs to target like when you want to stop the cell cycle if you need it. Okay, so going a little bit into cyclins more. So cyclins again give CDK binding specificity. The cells produce cyclins when they need it, when they're needed. So different phases within the cell cycle have different cyclins. So for example, in the case of this picture, for example, like M phase would have the cyclin B's produced. These are just like, for in this case, this example. Sometimes in exams or when Laura writes them, she'll just say like M phase cyclins or like S phase cyclins, S phase CDKs. She'll just say like what phase it is. So you don't necessarily have to know which letter goes with which one. So, and then just something to notice is that CDK, we just talked about like the 3 different ways that CDK can be regulated, or a bunch of different ways that CDK can be regulated. Cyclin is only controlled by the level of transcription. So they're transcribed. You can up the transcription or you can down regulate transcription, and that'll make more or less cyclins. And then you can also go through ubiquitin mediated proteolysis. So that just means like it gets ubiquitinated and then they get sent off to the proteasome to get like chopped up or whatever it is, eaten basically. Yeah. Okay. So bringing the CDK and the cyclin together, again, CDK alone, this activation loop, so this like activation loop is what it's called, that little red thing. It's like hidden when it's not with cyclin. Without your activation loop, your CDK is not active. So you have to have cyclin bound to the CDK in order for the entire complex to be active. So then once you have CDK and cyclin bound together though, it's still technically not like 100% active. So CDK has an CDK activating kinase, so that's CAC. A really big mistake on like the past exam was people were getting like MCAC and CAC confused. They're just completely different things. MCAC is like something that's not inside at like the centrosomes, or no, the centromeres, sorry, at the centromeres.
MCAC is kinesin 13.
Yeah, so it's like not related to cyclin CDK complexes at all. So just make sure you like know the difference and don't make that mistake again if that comes up. It was just a big mistake. I wanted to bring it up. So then, so CDK activating kinases, the CACs, they add phosphates to the CDK activation loop. And then that further allows CDK to be like more active. And then it can go phosphorylate whatever target protein or substrate is needed. And then like we said about before, another way to regulate CDK is the CDKI or the CDK inhibitor that blocks CDK from phosphorylating downstream proteins or substrates. Any questions about like CDK cyclin complex? Yeah.
I have a question for the exam.
Okay.
Can I ask that?
Yeah.
So that there was a question asking about if M-cyclin production was impurity stuff. And in my answer, I thought I wrote enough for detail wise, but they asked me if it was more relevant.
If you want to,
Yeah.
if you want to come up afterwards, we can
because I feel
specifically
like
look at it.
sometimes exams are a little bit more specific depending on like what each person wrote. So you can come back at the, if you have time, like at the end of this. Yes, you can ask and then we'll like look at it. Is that okay?
Yeah.
Cool. All right. All right. So we'll just keep going into cell cycle arrest. So cell cycle arrest has to do with P53. When your DNA is mutated, when anything is wrong within the cell cycle process, it's like a big red flag for the cell and it wants to obviously stop the cell cycle. So that's why it's like arresting it. So for example, if your DNA is mutated in G1, what's going to happen? Your P53 gets activated. So P53 is a transcription factor and a tumor suppressor. More specifically, it is a transcription factor, so then it can act as a tumor suppressor. And it does so because it responds to DNA damage. So it's a transcription factor. Once the cell realizes that DNA is mutated or something's wrong with the DNA, it'll be upregulated and thus it'll transcribe P21. And P21 is a CDK inhibitor. So bad DNA, then you get your P53, which is a transcription factor. That'll go upregulate the transcription of P21. And then P21 is a CDK inhibitor. So then that's going to inhibit CDK from doing its job. And like we said before, if you prevent CDK from doing its job, the cell cycle can't move forward. Yeah. If in this case, or in another scenario, if DNA damage is like completely irreversible, so it instead of just like stopping it and arresting it for now to fix whatever damage, if it's like beyond repair, it's a hot mess, it's just going to send it into apoptosis and get rid of the cell completely because it would rather not have it than put in that extra effort and energy into fixing something that's not worth it. Yes. All right, so
Sorry,
just
I'm on the YouTube channel. I'm not.
what?
I'm not just playing on my phone. I'm on the YouTube channel, making sure everything was uploaded.
You're good. So a couple of quick questions. Why is P53 a tumor suppressor or known as a tumor suppressor? Yeah, we'll go with you. I haven't heard from you yet. Yeah, exactly. Perfect. So P53 eventually causes P21 and P21 stops CDK. If you stop CDK, you stop the cell cycle. If you stop the cell cycle, you don't get cell division. And then that's a tumor suppressor because tumors are formed by a lot of cell division. Nice. So what happens if, on the other hand, like P53 is not available at all? Any ideas? You look like you want to. Yeah, go for it.
I know it's not.
Exactly. Yeah. So you could have a lot more DNA damage. If you have a lot of DNA damage and then nothing's happening to prevent it, you're going to continuously have that like bad cell growth. So then eventually you can become cancerous as well.
P53 is a big one in cancer. We love P53 in cancer.
Yeah. And I feel like a lot of time in her lecture, Laura sometimes like glosses over it, but then like emphasizes it in like exams and stuff because it is really important. So it's just important to like know about it. Okay. So, so the G1 phase, just kind of like a little bit of an overview of it all so that you guys have it all in like one area. So in its entirety, again, like a little summary, G1, just make sure that there's a lot of nutrients, that the cell is big enough to continue going on, that there's enough cellular machinery, so like proteins, enzymes, whatever it needs to go through with division. And of course, that DNA is not damaged. You can kind of split it up into like early, mid to late G1 phases. So in the beginning of G1, your G1 CDK complex starts to form, but obviously they're not active yet because you don't have enough CDKs. So when you have that low G1 CDK level, your pre-RCs form. You want to check it? Yeah. So your pre-RCs form, also known as the pre-replication complex. Sorry, that was the Zoom.
Don't worry.
Yes. So after your pre-RC forms in the mid to late G1 phase, this is where you have that restriction point or the point of no return. After the CDK becomes active, the cell has to pass this restriction point in order to move on to S phase. And then there's a slide going over like restriction point, RB, E2F, all that. So there's a little more detail about that in a second. And then lastly, in your late G1 phase, you have the pre-IC. And then this is when your CDK activity is a lot higher than it was in the beginning. It brings in a lot more of those extra proteins to make that pre-IC complex. Okay, so starting with the early G1 phase, your pre-RC, again, it forms when CDK activity is low. So that's the most important part. You have a couple of different things, a few different things happening in order to form the pre-RC. So your ORC, or the origin recognition complex, starts by beginning to bind onto the origin of your DNA. So if this green area is the origin, your ORC comes in and binds there. ORC then recruits CDC6, and then ORC and CDC6 recruits CDT1 and MCM. MCM is just another, like the more specific way to say what a helicase is. And then multiple MCMs can be recruited in order to unwind DNA in both directions. You can have one or multiple. You need at least two because you have them on like both sides. So altogether, these proteins, so your ORC, your CDC6, and your MCM, they all come together to form your pre-RC. So those are like the three main parts of your pre-RCs, which is important for later on when we talk about like S phase and like what needs to be gotten rid of in order to go into S phase. You need to get rid of those three specific things, because if they're there and present, then you have the chance of making a pre-RC again. All right, so then into the middle of, or like mid G1 phase, your restriction point or the point of no return. This is when the cell, sorry, commits to the cell cycle. Kind of like, you know, we want to go through with this, no backseas type of thing. So when everything's in check and it makes sure that the cell's big enough, enough nutrients, all that stuff that we said in the beginning, the G1 cyclin CDK is going to become activated, and then it's going to be able to phosphorylate RB. So this big purple blob protein. RB is initially attached to E2F. When that's the case, E2F cannot go do its job. However, when G1 cyclin CDK goes to phosphorylate RB, it can let go of E2F. So now that E2F and RB are separated, E2F can then go and transcribe the proteins that are needed for S phase. So like it says on the slide, it's kept inhibited by RB until there's enough G1 CDK levels. And that's, again, just to make sure that the cell is big enough and like ready to continue moving on. Once it is ready to move on, E2F separates from RB because RB was phosphorylated, and then E2F goes to transcribe the proteins that are needed for S phase. Those proteins are then going to be used for forming the pre-IC complex. So the pre-IC complex is just a larger complex, the pre-initiation complex. So you go from the pre-RC, which was only like those 3 proteins, to the pre-IC. Once all those proteins are added onto the pre-RC, you get your pre-IC, and then eventually DNA polymerase also has to get loaded on. Obviously, because if you don't have DNA polymerase, you can't actually have your DNA replication process. And then once DNA replication starts, you are going into S phase. So that's like origin firing. You're firing into S phase. Something that's just like good to note is that you can have like multiple firings going on. They don't all have to happen at the same time, but if you have like a long strand of DNA, you can have like multiple different pre-RCs and then multiple different origin firings that can happen at the same time. They can happen at different times, but it doesn't really matter as long as you know like what's going on at one. So these are just some quick questions. If P53 is suppressed or damaged, what happens to the cell? Oh, we already answered that kind of in the beginning. If P53 is suppressed or damaged, the cells could become cancerous or like you had a lot of DNA damage. So what about number 2, true or false? In response to DNA damage, cells should increase concentration of CDK inhibitors. True. Yeah, there you go. If you increase CDK inhibitors, that means that CDK is not working, or at least temporarily not working. So then your cell cycle stops and then it can work on fixing the DNA damage if needed. For 3, true or false, in response to DNA damage, E2F is active. False, exactly. Do you want to explain why? Yeah.
Basically, in response to data,
Yeah, exactly. So just for the recording, E2F should not be active because if E2F is active, it's going to continue on to S phase and to the next phase of the cell cycle. Number 4, could E2F be a cause of cancer? Can a cell divide if E2F is not functional? So like if you have too much E2F, can cancer happen basically? Yeah, exactly. So same reasoning, E2F causes like continuation of the cell cycle. So if you have too much E2F, you potentially could have like too much cell division. And then last one, true or false, pre-RCs can form at the end of G1 after CDK is fully activated. False. You want to explain why it's false?
Me?
Yeah, sorry.
Doesn't the pre-IC
So actually the opposite, pre-RCs have to happen first and then the pre-ICs happen. But the reason that it's false is because pre-RCs can only form when you have low CDK levels, because once you have high CDK levels, then you get your pre-ICs, and then everything that makes the pre-RC gets phosphorylated, and then they get kind of like shoved away from anywhere inside the nucleus to make sure that pre-RCs don't form again. So that is a perfect transition into the S phase. So like we just covered before, you fire into your S phase because your pre-ICs formed and you have your origin firing, everything's all set with your DNA polymerase. So once you're in S phase, then you also have active SCDKs. The active SCDKs, as I was saying before, they suppress pre-RC reassembly by phosphorylating the ORC, the CDC6, and also the MCM. So those 3 things, to reiterate, are what make up your pre-RC. If you phosphorylate those 3 things, they get different things happen to them. So your prevention of CDC6 binding, degradation, or nuclear export, essentially doing what needs to be done to make sure that they're not around in the nucleus to form again. Because if your pre-RC forms again, you have a potential chance of firing DNA replication again, and you don't want that, because if you do multiple DNA replication firings, that increases your chances of DNA replication damage. Does that make sense? All All right, so S phase specifically, you have your cohesin rings that form around replicating DNA. The rings make sure that DNA strands stay together throughout replication and duplication so that you don't get like in the end, you don't get cells that have a different amount of DNA in the different cells. They prevent chromosome segregation, and they have to be present in S phase in order for M phase to function. Again, because of the whole, like, you don't want the chromosomes separating before they need to be. And then there's also S phase checkpoint, which is basically just checking if there is DNA damage, is all of the DNA replicated completely? And if necessary, similar to before, the cell will put it into cell cycle arrest if needed, so that your, so that your cell cycle stops if needed by P53. Okay. So then G2 phase, your cell is growing and prepping and just making sure that it's big enough. So specifically, it does that by the G2 checkpoint. So at this point, your mCDK levels are slowly rising as it's getting closer and closer to M phase, but it is still inactive because we're still in G2. So what happens is your mCDK is around, and then M cyclin comes in to bind to it. But as I said, like right in the beginning, even though your cyclin binds to your CDK, it doesn't necessarily mean that it's active yet because it has to go through phosphorylation as well. So it gets phosphorylated twice, one by WEE1, which is inhibitory, and then one by CAC, which is activating. When you put an inhibitory and an activating phosphate together, the inhibitory one is more powerful essentially. So that means that it's going to like overrule the activating one and prevent CDK from being activated. So then that means that your cell cycle is going to stay in G2 for now. It's not going to go into M phase yet. And so that just means that it wants to make sure that the cell has enough time to grow bigger. So if your WEE1 isn't functional, if it's not there, chances are you're probably going to have a smaller cell. Yeah. And then on the other hand, like if your WEE1 is there for too long and it never gets removed, chances are you're going to have like a bigger cell than you normally would have because it was in G2 for too long. So that's kind of like the coin flip. After your CDC25, which is an activating phosphatase, is activated, it can go and take off. Sorry, the two laptop thing confuses me too. It can go and take off the inhibitory phosphate so that now your entire M-cyclin CDK is active because all it has is this one activating phosphate. However, it's not like at its 100% potential yet until it goes and does the positive feedback loop. So your active mCDK is going to go back and activate more CDC25, and then that's just going to do more active M-cyclin CDK. So that's why it's a positive feedback loop, because the like the product of this loop is making more of the same product. Does that make sense? All right, cool. We're almost there, guys. All right, so just a few questions again. What if there was a gain of function? So that means like it was like overactive basically in CDC25. Yeah. So gain of function means that it can do, yeah, not loss of function. No, that's okay. If it was loss of function, that's right. So then gain of function is just the opposite. If you had a gain of function in CDC25, it's just gonna like, yeah, so it would probably take off the WEE1 phosphate early, or like maybe even prevent Wee1 phosphate from putting it on at all. And then your cell will be probably a lot smaller going into M phase. Yeah. What if Wee1, on the other hand, had a loss of function mutation? Yep. Exactly. Again, it would probably be too small because it would only have the activating phosphate and go straight into M phase. Didn't have enough time to grow. On the other hand, if Wee1 had a gain of function, it would be probably too big because it's like stuck in G2 for longer than it has to be. Yeah.
Is it possible, like, for that question in my brain, like, if we want to have a data function, it would never come off? Is it okay to answer it like that? Or is it better?
So,
Is that for my brain?
so Wee1 is what's putting the phosphate on.
Yeah.
So you want to go with what Wee1 does, not with what like Wee1 puts on. So what you're thinking is like, oh, what if that inhibitory phosphate had a gain of function?
Yeah.
Then that would mean that your cell's going to stay there and it's going to become a lot bigger. But if Wee1 had a gain of function, that means like Wee1 itself is able to inhibit more.
Okay.
Yeah. Does that make sense? So you want to make sure like you're talking about what Wee1 is actually doing rather than like what the end product is.
Oh.
Wait.
So, like,
Oh, maybe I misunderstood your question.
yeah, I think she was thinking, I was thinking.
Okay.
The Wee1 itself is gonna keep trying to inhibit it, correct?
Mm-hmm.
But the phosphate itself, like if you look at it, that's what's staying on.
Yeah.
Keeping the phosphate there is just creating, making that phosphate happen.
Oh, I see what you mean. Okay, yeah. So if you answered it in the sense of like, oh, Wee1 would stay on for a while, it's not wrong because we just don't know for sure, like in a cell, if it's like one single Wee1 or if it's like many Wee1s. But as long as you answer in the sense of like this cell's not going into the next phase, that would be correct. Yeah. Does that make sense?
Yeah.
The wee one.
All right. Cool. So then
We're getting near the end. Don't worry.
this is
Just hang
yeah,
in.
we're
It's the
getting
last-
there.
It's the home stretch.
So lastly, or last phase that has many mini phases, you have your M phase. So prophase, metaphase, anaphase, telophase / cytokinesis. This is kind of just like a very brief, like what each phase kind of does. So you can take a look at it on your own if you want. Going into prophase first, this is when you have your active MCDKs. So before CDC25 took off the inhibitory one, now your cell's in M phase. And so you have active MCDKs. Active MCDKs do like mainly 3 different things that are important to know. They phosphorylate lamins, which means like the outside nuclear envelope or the lamina gets broken down so that all your chromosomes on the inside can free float. At the same time, because now you can get to the chromosomes, it phosphorylates condensin, which makes the chromosomes condense. So they go from like spaghetti noodles into like more compact sausages. I don't know why I keep on using that
I hate
analogy.
it.
It's so bad, but that's what like sticks in my brain because that's how she teaches it. So it's just like, always stuck. Do what you will with it. Then it also phosphorylates various maps and motors in order to facilitate spindle assembly. So that was like everything that Emily was talking about before. Content builds up. All right. Some more main ideas that if you want like more specific details on, we definitely have like more details on that in like previous slides and lectures. But this is just like kind of what what you need to know the gist of. So sequential phosphorylation, phosphorylation has to happen in a specific order. If you phosphorylate something upstream, that's going to cause a downstream effect. If you don't phosphorylate that first thing that had to be phosphorylated, chances are what's downstream isn't going to happen unless you have like other crosstalk or things from like other places coming in. But for that one specific pathway, it probably will not occur. And so that's why like CDK is a master cell regulator, because if you prevent CDK from phosphorylating other things, you can most likely stop the cell cycle because everything downstream from the CDK isn't going to be active. Your spindle assembly, everything. So naturally your cell has mitotic spindles already. So it has like microtubules, sorry, not spindles. Naturally your cell already has microtubules, but you want to break them all down and then make them into spindles so that it can start to go into M phase. An example that she's talked about is katanin, where it basically just like cuts it up and just like everything catastrophes, they all depolymerize. And so once they all quickly depolymerize, they can again quickly polymerize and reassemble into spindles so that it can be ready to separate the chromosomes in anaphase. Your kinetochores are important to know because they're the large protein complexes that are keeping your chromosomes together. It's at the centromeres of the sister chromatids, making sure that they stay together until they need to be separated. This is where your spindle microtubules attach to, where you have like different motor proteins and stuff that are like going to like help separate it. And again, the opposing forces idea that Emily talked about before as well. Your spindle formation requires opposing forces in order to stabilize them. So that push and pull mechanism of like all the spindles, where that it's stabilizing the cell as a whole and then eventually going to pull everything away from each other and push everything away from each other as well. Oh, it is here. This is the slide this is the picture I was talking about, where it's like a white like textbook image and it looks like bunnies. I don't know. Do they not look like? Okay.
This is being recorded. I'll talk to you later.
Okay.
Oh, those ones.
Yes.
Okay. A little bit.
All right. So anyways, the white ones are dynein. So they're negative, or sorry, not negative, physics. They are minus n directed, where they walk towards the centrosomes, and then they pull the poles apart from each other. Whereas in the other ones are, they're all dyneins, but the white ones specifically are minus n directed. And then the black ones are plus n directed. So that's what the arrows are showing. And then this is like a little drawing that I think was just on Laura's slides before as well. So it kind of shows a little bit more about like which way's plus, which way's minus, how they push and pull away from each other.
I think it's really helpful if you like get stuck and don't know what's happening. If you like draw a plus and a minus on a piece of paper and then like walk your fingers and see which way the paper moves. That's what I used to have to do.
Oh my gosh, that's really smart.
Yeah. So like if it's walking towards the plus end, walk your fingers on the paper and it'll like move towards the minus end. Like the minus end will like push further towards you. It'll make sense if you do it.
There's also more like speech notes of like what the push and pull model actually does in the notes of this like slide. So then you can like take a look at that too later when I send the slides out.
Good luck.
I hope it works.
Sending the slides.
All right,
PDF.
going into metaphase. So this is when your sister chromatids are still secured by your cohesin rings from before. That was done during S phase. They are lined up at the center of the cell now. Your microtubules are connected to the kinetochores of your sister chromatids, and then the cell can basically just kind of like sense this equal opposing tension, because theoretically it should be in the middle of the cell, so it's going to feel equal tension on both sides. And once the cell feels this, they realize that, okay, everything's lined up in the middle. Let's move on to the next phase. The next phase would be anaphase, but in order to get into anaphase, you have your spindle assembly checkpoint. So this is during / a little bit after M phase, once the cells are all lined up, and sorry, not the cells, once the chromosomes are all lined up, in order to separate from each other, because they still have the cohesin rings, they have to be broken apart. Otherwise, your chromosomes are going to be attached continuously, which is not what you want. The way that your cohesin rings get degraded are that CDC20 is released and it binds and activates APC. So CDC20 goes to activate APC, which is like this blue teal color one. Once your APC is activated, it acts as a ubiquitin ligase. And what we know about anything that gets ubiquitinated is that it gets broken apart or, just like kind of chopped up, cut into pieces. And so your CDC20 APC complex will then go and target securin, which is this like orange crown looking protein. And once securin is targeted, it'll go and separate from separase. So originally, securin and separase are bound together, but then APC CDC20 complex goes and ubiquitinates securin, and then they are no longer attached to each other because securin is ubiquitinated, chopped up into pieces. All you have is a solo separase, which has changed conformation because it's no longer bound to securin. And then it can go and cleave all of the cohesin rings, which are like the pink rings that are shown at the bottom. Cleave the cohesin rings so that your sister chromatids are separated from each other, and then they could go into anaphase. Any questions about that? Okay. Just like a little side note, so APC, because we're nearing like the end of the cell cycle, so APC also targets M cyclin for degradation. So this is when your M cyclin CDK starts, the levels of it starts to drop again because you're nearing the end of the cell cycle where your CDK levels are low. So that's kind of all happening at the same time. All right. Anaphase, you have anaphase A versus anaphase B. Anaphase A is when your chromosomes are pulled poleward. So you want to think from like from the middle, the center, and pulled out. Chromosomes can be walked on or like walked outward by your dyneins or any minus N directed motors. Your kinetochore microtubules can also shorten, or your spindle poles. Your spindle poles stay in place here. So anything that regards your spindle poles being pulled apart, though, that is anaphase B. Your spindles can lengthen, so that's going to push the poles apart. Your sliding forces between the interpolar microtubules can also cause the poles to be pulled apart. And then just any pulling force that's directly on the poles as well. So any of the ones that are on like the aster microtubules, which are these like outside ones that are connected to the cell membrane, if those are being pulled at, that'll also pull away the spindle poles. So anaphase A and anaphase B, they're separate, but they do both occur. They don't necessarily have to occur like A first then B or B first then A. They all kind of happen at the same time or at different times, but they both do happen. So that was a question that a lot of people have. All right. So now now your sister chromatids are separated, they're pulled apart from each other, and you have to go into telophase and cytokinesis because that's the last step for actually getting two separate daughter cells. It happens by a cleavage furrow that starts to form, which is basically like a ring that deepens and like pushes in the middle of the two membranes until it hits the spindles in the center. And just basically like snaps the two in half, and eventually they split and you get two separate daughter cells. And remember before as I was saying, the APC levels were also causing CDK to drop. And so once your M cyclin CDK, originally M cyclin CDK was causing your nuclear envelope to break away, was causing your chromosomes to become more condensed and like more solid. And it was also causing a bunch of like motors and microtubules and that formation for the spindle assembly. So now that your CDK levels have dropped, the opposite is going to happen. So your spindles are going to disassemble, your chromosomes are going to decondense, and then your nuclear envelope is going to reform around all of this new DNA that has happened. And then that is the end of the cell cycle because you're low CDK, you have the potential for pre-RCs to form again. And if you have the potential for pre-RCs to form again, that means you can replicate again. So it's like this whole cycle. All right. Last few questions, just to kind of get those brains churning a little more. What if you have a loss of function in your APC? Any ideas? So APC was the one that could cause ubiquitination. think again, is it the spindle poles that can't separate, or that's okay. Yeah, so your sister chromatids can't separate because it won't be able to cut any of the cohesin rings, right? Nice. Yeah, exactly, because M cyclin and CDK are bound together right now. If you have high M cyclin, usually you have high CDK as well, since they're bound together. If you can't degrade M cyclin because there's a loss of function in APC maybe, then you're gonna continuously have like active MCDK and you won't be able to restart. Yeah. All right. What if the cell only had minus N directed dyneins for spindle assembly? So only minus N, no plus N.
Think of the bunnies.
Let's ask this. Do you need both minus N and plus N directed in order for that push and pull function to happen? Okay, so if you need both push and pull for minus and plus ended, then if you only have minus, then is your cell going to be able to like properly form spindle assembly and pull apart? Exactly, yeah. Because you have, again, you have to have that like right perfect tension for the cell to realize when it's ready to go on. If you only have one force and not the other, it's going to be like imbalanced and the cell's never going to get to that like middle ground that it needs to be. What if cohesin cannot be cleaved? So similar to the first one, but a little more direct.
You raised your hand.
Okay. Yeah, exactly. So same answer. Sister chromatids can't form apart. You don't get anaphase. What if securin has a loss of function? Securin is bound to separase. So if securin is not doing its job, what separates? Yeah, exactly. You would move on to anaphase before maybe it's ready to move on to anaphase because like there's nothing holding the sister chromatids together. So it's a little bit unsure. Or the cohesin might not have been able to bind in the first place because that's what's securing them together. Yeah. Thank you. Number 5, what if dyneins on the aster microtubules? Oh, oh, sorry.
It took me a minute
I didn't know
too.
what the question was asking. I made these questions, I promise you. Dyneins that are on the aster microtubule. So this is kind of questioning if you know what dyneins are, if you know what aster microtubules are. Does that consist of anaphase A or anaphase B?
Are the dyneins on the aster microtubules an A or B? Go back a few slides.
We can.
No, not on that one. Oh yeah, on both.
Oh, right.
Anaphase A or anaphase B?
So the aster microtubules are like these like star ones that are outside. Oh, what were you gonna say? Oh, that's okay. All right. So think about it like this. If it is on the aster microtubules and they're minus N directed, which way are they walking? Towards the centrosome or towards the edge? So minus N is the centrosome. There you go. Minus N is the centrosome. Just remember that, you know, the minus N is where like there's not a lot of growth or shrinking happening. So if it's like embedded inside the centrosome, you can't really grow or shrink from it. So that's the minus N. So if it's walking towards the minus N, it's like pulling it out. Like if it sounds like weird and it doesn't feel right, but if it's walking towards the minus N, it's pulling it in the opposite direction essentially.
Think of the paper. Yeah, so if you walk, I'm so sorry for the people on the video who aren't gonna be able to see this right now. but if you, so say that this is your minus end, right? So if I'm this isn't gonna move the way I want, but if I'm walking towards my minus end, the paper's going to go, like the plus end gets lower, right? So if
Yeah.
I'm walking towards the central zone, I'm pulling the microtubule further to the edge of the cell,
Mhm.
right? To the, or the, you know what I'm saying, right? Does that make sense? Yeah.
Yeah. So then
I
you're,
mean, that, I got it, but.
Yeah.
Yeah.
Yes, it does.
yeah.
Sorry
So then
for
you're,
My hands get doubled out.
exactly. So then your spindle pole is getting pulled further out. So if it's your spindle poles that are moving, is this anaphase A or anaphase B? There you go. Yay.
Woo. Yeah, it's
Tis
B.
B.
For those on the recording that couldn't hear that.
Tis B.
Tis B.
All right. So on the other hand, if you have dyneins, so again, minus N directed on the kinetochore microtubules, is that A or is that B?
Say it with confidence now.
There you go.
A.
You want to
Woo.
explain or try to explain? Yeah.
Yeah.
Yeah, exactly.
Want to say that again for
So,
the recording?
so if it's walking towards the minus end on the kinetochore microtubules, you're going in the opposite direction. So it's like pulling the sister chromatid along with it away from each other. So because you're splitting apart the sister chromatids, that's anaphase A. All right.
This is really hard. I'm proud of everyone.
Yeah.
Like anaphase A and B is definitely really confusing, and walking microtubules in static pictures is definitely hard.
Yeah.
And on that note,
So these are just little videos. Yeah. Again, like for your own convenience, if you need like different ways of learning, different people talking to you, that's not us. These are here for your convenience. People have found them useful before, so we just kind of put them there. It's definitely going to play. Please. Hang on.
just
Okay.
keeps like all night.
No.
The only way.
No. Okay. Oh, okay. Another video again for your convenience.
An adult human contains an
An adult human contains an
Overstimulated.
estimate.
I
Okay.
want to know what an adult human contains now.
So
I'm gonna go watch it.
yeah. You want to wrap it up?
That's a wrap. So thank you. This is definitely a long one, but this is definitely some dense material. I didn't pick these memes, so if they're not funny, that one's not on me.
I think so.
No.
These are old slides, man.
So as always, thank you for attending and good luck. Please contact us with any questions. I know that a few of us are active in the Discord, and if not, feel free to email us. You're gonna get the quickest response if you CC everyone. Yeah, thank you to those in the recording. Okay.
Yes,
You stopped
I'm
recording.
gonna
Oh
stop.
no, you did stop recording. Okay.